Okay, let's go ahead and get started. So I guess maybe one of the most important things is to, we're going to have a party on Wednesday. Everybody's invited. Go ahead and tell your friends who aren't here today that they're invited also. So it'll cover chapters 6 through 10, which we finished last week. We started chapter 11, which begins the material for exam 3. I'll probably get most of the, might finish chapter 11, or at least get most of the way through it. But what I'll do is I'll stop about 15 to 20 minutes before our normal time, probably around 20 after 3, something like that. And then I'll open up the floor at that point for questions related to the exam. The other is it'll probably be helpful during that 15 or 20 minutes. I'll run another one of the roll calls for the eye clickers. So if I understand the system correctly, if your name is not up there, and the list should be getting pretty short now, and you've been using an eye clicker, then you have an eye clicker registered. And then shortly, maybe around the midterm or so, I'll publish another list of the registered eye clickers so you know you're good to go. So the exam will be same ground rules, same format as last time. We'll have several proctors here. No calculators, no cell phones, no electronic devices. Come in with the pencils. Look at the Scantron. I think the how to fill it out is still up on the web, up on Blackboard. Fill it out ahead of time. You got to fill in those little circles, okay? Do everything you can to look around you. There'll be a lot more people here on Wednesday. Do everything you can to facilitate my accurately recording your grade. Let's see. No questions as we did last time because we can't really get into the center aisles, but you'll be able to write on the exam. I've put another. Did that help last time having the blank page on the back? Okay, so that's on there this time. So again, format should be the same. You know the drill by now. Any general questions? Okay. So before we get what did I do? Let's see how fast you work. All right. Try to answer the question. Okay, you saw I got carried away there. So, from last time, this idea of supercoiling. What the hell's going on? What am I missing? Or are you just laughing because I showed you the answer already? All right. I just want to see how fast your reflexes work. Try to answer the question without, and then I put the right answer down. This is here to help you understand this idea of the supercoiling. We're going to go over it for a couple minutes before I move on to the new material. But we're going to stop at 1 minute since half of you are going, what was it again? And then the more important question is, did I get it right? All right, so we're going to do it, we're going to go to 1 minute. Okay. So what's the answer I showed you? Oh, I just have to go back to, for some reason I have to do that first. What, you didn't even see it? Okay, is this the right answer? Don't know. Okay. So last time we did one of these problems. So we have supercoiled and we have positive supercoils, negative supercoils. As we're going to go over in a minute, if it's over-rotated, if you have more turns, more rotations than you should have, it is what? That gives you positive supercoils. If it's under-rotated, you get negative supercoils. Now, I didn't give you the size of the molecule here. I just told you there were 8 base pairs per turn of the helix. What's the normal number? 10. That's what it says up there, right? So if you have fewer base pairs per turn, then you'd have to have more turns, right? Regardless of the size of the molecule. So now it's over-rotated. There's more turns than should be there, and therefore it would give you positive supercoils. Okay, the last time we did the problem, it was 300 base pairs, and I think it said it had 20 turns. How many turns of the helix would you have with a relaxed circular molecule of 300 base pairs? 30. So there it was under-rotated, and you had negative supercoils. Okay. So we'll do one more question for what we're going to talk about today, then we'll go back over that real quick to show you those diagrams. I probably shouldn't have done this because you guys have to key it all in, but let's see. So I have to go to here. But the word's not that long. It's not working? What parts?
It's on the ABCD, it's not on the letters. That's not for alphabet. But it's trying to do.
What a nuisance. What a... Or didn't it...
Yeah, they're...
Or didn't... Oh, I guess it didn't change. Okay, there. It didn't change. That's what the problem was. Okay. That work? Is this some sort of distress signal? So I hear this whole hum kind of. Alright. Looks like you're going to need about 2 minutes. The rate it's going. Can you imagine typing your dissertation this way? Be here for years.
Make a spelling error on things.
There was probably no spell checker built in, right? Okay, so as I said, we'll go to 2 minutes on this one. Okay. 3, 2, 1. All right. So what's the word that took so long to spell? Okay. And we don't, that's why we don't do many of those. Yes? I'm sorry, what? Yeah, it shows me all the responses. So histone, histones. You said non-histones, that's wrong. Okay. So as I said, this is chapter 11. So what we've really been doing is looking at the different structural levels of DNA. So in chapter 10, which you should be very familiar with by now, you had the primary sequence, which was the nucleotide sequence, which really is the primary source of encoded information, the genetic information. The secondary level was that double-stranded structure that we talked about. And in this chapter, we're especially focusing on eukaryotic cells. We're going into this higher level coiling that you find in chromatin. So just to review quickly what was really the basis of that first question. So this is what we're talking about. So you have this torsional stress when you have a circular molecule. So everything is built around this 10 base pairs per turn that you have in a relaxed circle. So here was the example we had before, and you can see the electron micrograph. And then there's torsional twisting. If it's over-rotated, you get the positive supercoils. Under-rotated, you get negative. the outcome is really the same. It just is the direction in which the supercoils go. Positive supercoil, so the standard B-form DNA is a right-handed helix. Remember we said you could put your thumb down from your right hand in the natural direction your fingers curve is the direction of the helix. Positive supercoils flip in the same direction, that same right-handed direction. The negative supercoils just flip in the opposite direction. Now, in terms of what occurs naturally in cells, the DNA is negatively supercoiled. Okay, we also talked about topoisomerases. We're going to see numerous examples as we talk about transcription and DNA replication and even DNA repair. Whereas the strands are unwound, the double-strand DNA is unwound, it creates superhelical stress ahead of that unwinding DNA, and the cell has to have some mechanism to deal with those superhelical coils, or otherwise it eventually gets so knotted up the system will just shut down. And so these enzymes, as a group called topoisomerases, their job is to either add or remove the supercoiling as needed by the cell. There's different categories of them. Mainly, I just want you to be aware of, as a group, these enzymes, what they do is they change the supercoiling, and they do this by literally binding to the DNA, breaking one or two covalent bonds, depending on which type they are, pass the strands around the other strand, and then reform the phosphodiester bond. So that's where we were. That's where we ended up on last Wednesday. So now just a few comments about the chromosome in bacteria, and then the rest of it will really emphasize eukaryotic chromatin. A major difference is the amount of protein associated with the DNA. In bacterial cells, you have this circular double-stranded DNA. With only a few exceptions, you have this one chromosome. For instance, E. coli has the one circular genome, and there's a small amount of protein associated with it. It's not protein-free, but it's a very, very small amount, especially compared to the amount of protein we'll see with chromatin in eukaryotic cells. It's also not just existing as this relaxed circle in the cell, but it has these negative supercoils. And so basically it looks like a clump over in one. It's usually associated with the cell inner cell membrane, and it uses the term nucleoid to refer to this sort of mass of the genomic DNA over in one portion of the cell. What you see here is an E. coli where it's been treated to basically begin to permeabilize the cell wall a little bit, and it's allowing the DNA to come spewing out of here. So this is an electron micrograph. And so all of this stringy material, this is just a fraction of all the DNA coming out from inside this E. coli cell. One of the major functions of those proteins that are associated with it is to basically constrain it into loops of DNA, and this helps in this packaging process. And in eukaryotic cells, we'll see that this occurs at a much, much higher level, but the same basic concept holds. Okay, so that's what we want to look at is with eukaryotic chromosomes. So when we talked about meiosis and mitosis back in chapter 2, we said, okay, during interphase, the chromatin is very diffuse relative to its highly condensed state that you see of these metaphase chromosomes. Okay, that was a little bit of a simplification. Even during interphase, there's still a lot of condensation and supercoiling and compaction of the DNA. It's just less. So there's far less of it, but there still is a lot of this organization and this compaction. And much of it is going to have to be shifted and moved around as the genes have to be expressed. Okay. So at the first level, we can kind of divide this chromatin into two structures, euchromatin and heterochromatin. Euchromatin is going to be the less condensed, more diffuse parts of the chromosome. And this is where most of the transcription is going to occur. So we'll talk about it in a little more detail a little bit later. But the cells, in order this transcription, this whole RNA polymerase complex has to get in. It has to get access to the DNA, and it can't do that if the chromatin is in a very tight conformation. So this Euchromatin is the more diffuse, lighter condensing areas, and this is where we think transcription is primarily occurring. The heterochromatin is the more densely compacted parts of the interphase chromatin. So if you, I don't have a picture of it, but some of those old karyotypes, let's say where you see the metaphase chromosomes and it's stained with the DNA, a dye that specifically stains DNA, and you see banding patterns on the chromosome, those darkly staining areas are the heterochromatin. So those are the areas where the DNA is most tightly compacted. But you even have this in interphase chromatin. One area you find it on all the chromosomes is around the centromeres and near the telomeres. So from most of the studies that are available, it looks like if there is transcription is active in the areas of the centromere and the telomeres, it interferes with their function. So most likely this heterochromatin is functioning to keep transcription quiescent around that centromere and around the telomeres. There are also other regions along the chromosome that are in this heterochromatic, non-transcribed region, and that really varies from chromosome to chromosome. The bar bodies that we talked about, the inactivation of the X chromosome in female mammalian cells is almost exclusively heterochromatin. So it's getting condensed down, being taken into a transcriptionally inactive state, and it's now dense enough, and the heterochromatin part of that bar body is compact enough that even if you look at it during interphase, you see this darkly staining body. This is what was called the bar bodies. So what we're going to see, though, is there's going to be this chromatin is dynamic, and there's going to be changes in this chromatin conformation depending on the genes that are being transcribed. Okay. So one of the major players in this whole organization of the chromatin are the histones. So these are the major proteins associated with the chromatin. They are basic proteins, which simply means they're positively charged at physiological pH. So they're rich in lysine and arginine. In fact, they're sort of at the terminus of the proteins is really where you have some additional lysine and arginine residues. And the whole idea is the positive charges on these histones helps to neutralize the negative charges on the DNA. So remember, we have this sugar phosphate backbone on the DNA. You have the negative charges. Those strands are going to tend to repel each other. Now, especially if we're trying to condense this chromatin and get it into a very tightly packed organization, we want to neutralize those negative charges. The histones are some of the most highly conserved proteins. They're fairly small proteins. I think histone H3 is around 60 amino acids. And if you compare the sequence of histone H3 from a pea plant to that of a cow, I believe they only differ by 2 or 3 amino acids. So if you're doing molecular evolution and that's all the data you had available to you, you would conclude that peas and cows are very closely related, right? So how you do this type of analysis depends on what protein you're looking at. Any guesses as to why the histones might be so highly conserved in eukaryotic? What does it suggest about their functions? Yeah, they evolved very early in the evolution of eukaryotic cells, and once the functionality was there, there was no real driving force to change it. So when you see these highly conserved, actin's a very highly conserved protein, tubulin, a very highly conserved protein, begins to tell you that these proteins had important functions in the cells. They were there from the earliest days of eukaryotic cell evolution, and they've been carried through the whole evolutionary process. So there's 5 major histones: H1, H2A, H2B, H3, and H4. And they make up about 50% of the protein associated with chromatin. So we got lots of 50%. Chromatin is about 50% protein, and about 50% of that protein are histones. All right. So the other 50% must be, has this very creative name, non-histone proteins. Okay. These are a little harder to define. They're a very heterogeneous group. The majority are associated with chromatin. Some are associated with the nuclear matrix, the scaffolding structure within the nucleus. So they're not strictly associated with the chromatin. If you do any reading on them, you'll find some, they're called HMG proteins, for high mobility group proteins. Any ideas how they get their name? Well, when you run them on gels, they were very fast. They have a faster than expected electrophoresis through gels, so they got named high mobility group proteins. So it's just this catch-all for all of these other proteins that aren't in this histone category, where you have the 5 nice discrete proteins. Okay, so now what we can do is start to look at this chromatin structure in a little more detail. The very fundamental unit is the nucleosome. So these histones are going to form a core. The DNA is going to be wrapped around it, and then we're really going to just keep. It's just like with the polymers in the DNA we've already in the proteins and everything else. This is a repeating structure, kind of a common theme you see through molecular biology. When investigators were first studying chromatin structure and they started looking at it under the microscope, under the EM, under the electron microscope, it got this name beads on a string kind of appearance, because that's what they were seeing were these nucleosomes, and then a little linker section of DNA, then another little nucleosome. So at least morphologically it looked like a string of beads. So as molecular biologists, you really wanted to get more to the structure, the ultrastructure of this chromatin, these beads on a string. And what you needed was some kind of a reagent, something that you could go in, use as a tool to begin to dissect apart the structure of this chromatin. So what was used extensively was an enzyme that goes by the sort of jargon term DNase. So the ACE tells you it's an enzyme. So these are enzymes that do what? Degrade DNA. Very good. Not bad. Okay. Now, you don't want to use something like a restriction endonuclease because they're sequence specific. You want to use a DNase, which doesn't really care about the DNA sequence. It's just if the DNA is there, it's going to degrade it. So this is typically an enzyme called DNase 1 from E. coli, but we'll just call it DNase for our purposes. So the idea was, let's take a sample of chromatin and we'll add just a tiny amount of this enzyme. The rationale being whatever DNA is most accessible to the enzyme is what's going to get digested first. At the other extreme, any DNA that's tightly associated with protein is going to be protected from the nuclease. So these were DNA sensitivity assays. So you're just going to take the chromatin and treat it with a very, very low concentration of the DNase. And when you do that, and you look at the result, what you got were 200 base pair pieces of DNA. And they were very uniform in size, telling you it was a repeating structure. That about every 200 bases, there was something in this chromatin that made it very accessible to this enzyme. And then if you add a little bit more enzyme, what you found was that piece of DNA got smaller. And eventually you got to the point where it wasn't shortened any longer, and the idea being that was the DNA that was so closely associated with the protein that it was now resistant to the enzyme. So these got the name nuclease protection assay. The basis of it being DNA tightly associated with protein is going to be protected from the nuclease, and the chromatin in the most open configuration, the loosest configuration, the most euchromatic form, would be the first to be attacked by the DNAs. It would be most accessible. So using this as a molecular probe to probe the structure, the ultrastructure of the chromatin. Okay. So here's basically what they were showing. So this is what was giving the beads on a string view. So when they looked at the chromatin under electron microscope, they see a little bead, then a connecting string, then another bead, another connecting string. The idea here is when you add the first amount of nuclease, just a small amount, it clips the DNA that's connecting the nucleosomes together. And so this, when you use the lowest concentration of the DNase, it tends to clip it near the middle here, and then you isolate the DNA and you find that this DNA associated with the nucleosome is about 200 base pairs in length. And as I said, it was a very uniform sized DNA telling you that these nucleosomes are uniformly spaced along the chromatin rather than you go 200 bases and then you go 600 bases, then you go 700 bases. A very uniform repeating structure. Then if you add a little more nuclease, okay, so this is what's the chromatosome, the structure that you get when you first treat it. Then if you add a little more nuclease, that 200 bases shortens down to about 145 or so. That's now removing the DNA that's between the beads and cutting it back about as far as it can. Okay? So by doing these type of assays, what they came up with, and that in a biochemical analysis of the nucleosome, here's the structure that you're looking at. This core particle, which is this chromatosome right here, is going to have 8 proteins. So this nucleosome has 8 subunits. It's got 2 copies of each of these 4 histones, H2A, H2B, H3, and H4. Histone H1 is not part of that nucleosome core, that little round globe-like structure. Rather, the H1 is bound to the DNA, right? But you're really it's at this point, right where the DNA is leaving the surface of the nucleosome and going to the next one, is where the H1 is associated. So it's not part of the nucleosome core, that core particle of 8 subunits. Okay. And what it does, it helps to hold that DNA. So the DNA is on the surface of the histone. On the surface of the nucleosome. All right. So there's this little fragment of DNA, the sequence of DNA. It's only about 20 base pairs or so in length. That's where the histone H1 is. So at the most fundamental level, you have the nucleosome. You have a little less than 2 turns of DNA. It's about 1.6, 1.7 turns. That's this smallest amount, about 145 bases. Then you can add on this 20 or so bases of where the H1 is associated. That gives you the chromatosome. And then each chromatosome has about 30 or 40 bases of DNA that link to the next one. So that when they were cut in the middle of that linker DNA, that's how you got the 200 base pairs. So you can build this up from 145 base pairs on the surface of the nucleosome, about 167 when you include the binding site for H1, that's your chromatosome. Then add on another 30 or 40 bases, and you have the whole repeating unit. Okay? All right. So let's try this, see if we're going to get away from this. Mm-hmm. So which of the following is false regarding heterochromatin? Now we'll go to 1 minute and 15 seconds on this one. All right. The answer? Okay. All right. Oh, okay. Got another one here. Here we go. Everybody starts to panic because it involves math. And we'll do 1 minute and 15 seconds on this one also. All right. So were you able to do this one without a calculator? Nobody will admit it anyway. Okay. So you would have 100. The whole point being there's 2 copies of each of the 4 histones in each nucleosome. Okay. Any questions before we look at higher order stuff? Yes. You had 100 nucleosomes. You break it down into the individual proteins. There's 2 copies of histone H4 for each nucleosome, so that would give you 200 copies of histone H4. Okay? All right. Okay, this structure I just told you about, the beads on a string and such, that's really, for chromatin, pretty much the lowest level of the organization. But there's higher levels of coiling. And so I just want to kind of walk you through this. And what you see is these higher levels of coiling. At one point, this is pretty much what you see in interphase, where you can't really see chromosome structure. And then as the cells have to enter into M phase for either meiosis or mitosis, this chromatin condenses even further. And the chromosomes, when they're at metaphase, are at their most highly condensed state. And that's where you see the characteristic shape of each of these chromosomes. Okay. So this higher ordered structure, we basically have those beads on a string, and the next level of structure is called a solenoid, which is really, the idea is that this beads on a string are now being coiled. So if you're one of those, if you live in one of those guard gate communities where the HOA police come around and make sure that your garden hose isn't visible or something like that. So you're very careful and you kind of just coil the garden hose up over in the corner. That's how I think about these solenoids. So there's about 6 nucleosomes per layer, but you kind of coil them up. So you make this little kind of hollow fiber that's about 30 nanometers in diameter. And what's going to happen with this is it's going to be, as I showed you earlier, you had these big loops of the DNA coming out of the bacterial cell when they're anchored in place with protein. You have a similar situation with these eukaryotic chromosomes. So you have scaffold proteins that are part of the chromosome chromatin structure, and they anchor this chromatin in these large loops. except these loops are far larger than what you see in E. coli. As you can see, they're in the range of around 20 to 100,000 base pairs each. So that's what this is trying to show you here, is this coiling so that you end up with this fiber, they like to call it a fiber, and it's about 30 nanometers in diameter. Okay. This is really, in these loops, this is the basic structure you see during interphase for the chromatin. The next levels of condensation are really there just to condense the chromatin into the chromosome structures for meiosis and mitosis. And so what happens is these can coil and condense further into what's called to a structure that's about 250 nanometers, and finally gets all the way up to about 700 nanometers wide. That's really the metaphase chromosome. So to kind of walk you through this, so here's the most basic fundamental structure. This is just the double helix. It's not even really showing any protein associated with it. All right, and this whole structure is about 2 nanometers. So this is what we talked about in chapter 10, the fundamental structure of B-form DNA, and the helix is about 2 nanometers in diameter. Then you have this, the first level of coiling is wrapping the DNA around the surface of these nucleosomes. This one shows you the position of histone H1, so this green or yellow or whatever color it is, that's supposed to be the histone H1. And so that's associated with the DNA right at the point where the DNA is exiting the surface of the nucleosome, and then it transitions over, and then you go to the next and the next and the next. And you have this structure is about 11 nanometers in diameter. Okay. From there, you now can build up this 30 nanometer fiber, which is that solenoid we were talking about. So it gets sort of wrapped up in this coil, sort of the garden hose kind of analogy. And this is pretty much the level of condensation that you see in interphase. Then as you go towards M phase, where the chromosomes have to be compacted into a very, very small unit so they can physically be separated through meiosis and mitosis, you have this 300 nanometers. And finally, at the highest level, let's see here, and then finally out here, this is the highest level of condensation, and that's where they look like the typical chromosomes that you see in a karyotype. Okay. Any questions? Just trying not to get confused with what's going to be on the exam. All right? I can't process yet. I got to do the other stuff. Okay. All right. The life moves on. All right. Anybody know what Polytene chromosomes are? Because you didn't read ahead, right? No. If you read ahead, you would know. And, okay. Giant chromosomes. Yeah, good. You can read. Okay. So the Polytene chromosomes are a special kind of chromosomes. They're not found in every organism. They have been used extensively in Drosophila, so it's another one of the reasons that Drosophila was a really nice genetic model, especially through most of the 1900s. It was big because these giant chromosomes, what happens, and it only happens in certain cells, so it's typically done in the salivary glands. So if you want to look, I'll show you a picture of these, what these Polytene chromosomes look like. And when you want to do this under the microscope, and especially if you had a rough day and you want to take your vengeance out on something, I've done this before. These salivary glands are in the larvae, so you just kind of yank them out. And, it's cruel, isn't it? It's a maggot, okay? All right. So these Polytene chromosomes are in the salivary glands. The reason they're so large, they go through a process called endoreduplication. What that really means is DNA replication occurs, but the cells don't divide. And they go through repeated rounds of DNA replication until you have about 500 copies of each DNA molecule for each chromosome. So instead of having one DNA molecule that you would have in a mammalian chromosome, you have 500 of them, all stacked together, and they're all in exact register with each other, sequence for sequence. So because when they line up like this, when you look at them under the light microscope, you can see a very detailed banding pattern. And so this was very useful for a lot of the early Drosophila genetics. The other thing they showed was they started to give some of the early hints as to how chromatin changes organization when genes become transcriptionally active. So it was really in these Polytene, working with these Polytene chromosomes of Drosophila that was the heat shock response was first seen. Anybody know what heat shock? We talk about heat shock. Okay. So it's a type of stress. And so the heat shock mammals is a ubiquitous response from bacteria all the way up through humans. Under environmental stresses such as heat stress, a specific set of genes gets turned on and those heat shock proteins are made to help protect the cells. It actually took a long time to figure out what these proteins were doing. It was a lot of people had studied this heat shock response. It must do something to help protect the cell. We now know they're in a category of proteins called chaperone proteins, and they're there to help protect certain key proteins from being denatured. But for our purposes, what was found was that if you're looking at these Polytene chromosomes, and the next figure, I'll show you a figure in the next slide, when you heat shock them, you got what were called puffs. There would be changes in the shape of that Polytene chromosome, and one region would become much more diffuse, and so the chromatin was in a looser configuration, and that was because this is where the heat shock genes were being transcribed. So these are the Polytene chromosomes from Drosophila, or at least part of them. So one, as you can see, these very detailed banding patterns, and once you've worked with them for a while, so there's 4 chromosomes, 4 sets of chromosomes in Drosophila. Early on, in fact, Bridges, who discovered non-disjunction, I believe was the first to begin mapping these Polytene chromosomes. So each of these bands was given a specific number, and eventually people were able to use these bands to locate where their genes of interest were. And again, it was easy to visualize them under the light microscope because they were so large. And because these 500 copies are all in register with each other, you get these nice clean banding patterns. The other thing that's unusual here is you're really looking at the 2 homologs synapse together. So this is very much the way they would synapse during meiosis, but this is an interphase cell. This is unusual. This is not normal. It just happens in these cells that produce the Polytene chromosomes. So they're pretty common in the Diptera or the flies. They're not in every cell in the fly. They're just in limited tissue types. These come from the salivary glands. In terms of what we're talking about is this chromosome puff right here. This area, which is much more diffuse, you can see, so the dye is staining DNA. So you can see it's much more diffuse here. This is these Polytene chromosomes have now been warmed up a little bit. So normally the flies, they like about 25 degrees C. They do okay till they get to about 30 degrees C. You get a little above that, they're getting stressed. So if these Polytene chromosomes are warmed up a little bit above 30 degrees C, what happens is you have this change in conformation of the chromatin. You get this more diffuse area because this is where the heat shock genes are being transcribed and ultimately will be translated to make the heat shock proteins. Okay. Any questions?
What about like 50 degrees?
They're dead.
Really?
Yeah. When I was in California, we were using an incubator that was actually maintained by the university. Well, I mean, all the stocks were in there. We had backup stocks in other places, but every morning you came in and there was a big thermometer on the front, and it's like if that thing goes over 30 degrees, we're in trouble. So, yeah, they start getting stressed and they'll yeah. Okay. All right. So this next section is going to probe into this chromatin structure a little further. We're going to still use this DNA sensitivity that we already talked about, but now we're going to be looking at specific genes. The question being asked here is, does the chromatin actually change conformation specifically to activate transcription of those genes? So in other words, are the same genes in one cell where the genes are transcribed in a different chromatin conformation than they are in other tissues and other cells where the gene is not being expressed? So that's the purpose of this experiment. So this is again another one of these DNA sensitivity experiments. So here's the overall experiment, kind of walk you through it, and then I'll break it down into individual parts. So here we're looking at specific genes, specific regions of the chromosome. This is being done with the globin genes. So what are the globin genes? What do they code for? Globin proteins. Okay. And the globin proteins are found, are they important? This thing called hemoglobin. Okay. So the globin genes were used because, for technical reasons, they were one of the first eukaryotic genes to be cloned. So we had molecular probes available. The globin genes are involved in production of the red blood cells. So you would expect them to be active in erythrocytes. Is that true? Okay. So why was this done with chickens instead of mammalian cells? Anybody know? Could you do this with mammalian cells? No, mammalian cells actually have a little quirk in which the mature erythrocytes actually lose their nucleus. So they were useful for studies of translation, so you didn't have to have new genes being expressed, but they're not going to work for this because the genome's not there. So this was done with chicken erythrocytes. The labeling of this, and the idea is, the globin genes are a family of genes. Humans have about 7 beta globin genes, all clustered together on one chromosome. So here we have 3 different globin genes. The U indicates embryonic globin, so it's going to be the first one to be made. And then here are 2 different adult globins with the superscripts D and A. All right. So to kind of walk through, so here's the question being asked. Does the chromatin structure change as the transcription of these genes change? So that's the question we're asking. So we start out in the early DNA. Here's the embryonic globin gene, the 2 adult globin genes. The key to this is right here. This little arrow that it's going to show, when it shows up, these are what are called hypersensitive sites. These are going to be the point on the DNA that is most sensitive to the tiniest amount of the DNAs. It's going to be the first part of the DNA that's going to be accessible to the enzyme. All right. So before we start hemoglobin synthesis, so in the first 24 hours that the erythroblasts are developing, they're eventually going to become the erythrocytes. There are no hypersensitive sites along this DNA. These genes are not being transcribed. Then, 5 days into the maturation, right here, right at the beginning of the gene for the embryonic globin, is a hypersensitive site. So this sensitivity, this hypersensitivity to the DNAs corresponds to this gene being transcribed at this point in development. But the adult globin genes don't have this. Their areas are not sensitive to the nuclease. What is that telling you? That this DNA is in a different conformation than this. The accessibility of the enzyme The DNAs can get to the DNA here, but it can't get here. Now, if you do the experiment again, but you do this when the erythroblasts are 14 days old, now you're expressing the adult globins. You lose the hypersensitive sites for the embryonic, and the two adult globin genes now become hypersensitive. Okay. So it's changed. You're using the DNase as a probe to look at the structure of the chromatin. So this is telling you that the chromatin is changing conformation as the expression of the genes are changing. And if you do this in brain cells throughout this whole period of time, should you see any DNase hypersensitive sites? Is globin expressed in the brain? Hopefully not. Okay? And that's what you find. So basically, as a control, you see that there are no. So the same gene sequences in the brain remain in this heterochromatic conformation. They're not transcriptionally active, and they're not accessible to the DNase. So these were some of the early experiments that showed clearly that this chromatin is a dynamic structure, and its condensation state and this transition between heterochromatin and euchromatin changes with development, depends on the tissues and which genes are being expressed in those specific tissues. Okay, you can save your applause till the end. All right. All right, so let's try this. Give it a little break here. All right. A minute. We'll go to a minute and 15 seconds. Okay. All right. So what's the answer? All right. So will it be less sensitive to DNase? No. Will it be located in regions of relaxed chromatin? Yes. That's the whole, that was what we just showed you with the heat shock puff, all the chromatin. Think about it mechanically. This enzyme complexes have to get access into the chromatin, so it's got to be a looser, more relaxed configuration. Will it be more sensitive to the DNase? Yes, because now the enzyme has access to the DNA. Will it be located in heterochromatin? Well, that's the opposite of B. The heterochromatin is the condensed. Yes, ma'am. Say what? For these experiments, yes. I mean, so you're talking about the term. Okay. Digestion seems to be a term that's commonly used with DNases. So if we treat DNA with restriction endonucleases, which are DNases, the term is used digesting them. You are degrading it. The process of digesting the DNA with the DNase leads to DNA degradation. Does that make sense? Okay, don't shoot me. I just report the news. But yeah, some of this, molecular genetics is as much as any other field is just loaded with its own jargon and terminology. Okay. All right. just as a quick comment about epigenetics, we're going to come back. There's a whole sort of to see how this field has moved very quickly. We're using the 2014 edition of the textbook, which is actually pretty current. Although there's a few things we'll talk about that are too new to be in this textbook. The 2012, they come up with a new edition every 2 years. In the 2012 edition, epigenetics was kind of sprinkled throughout the chapters like it is here. By the time we got to the 2014 edition, there's a whole chapter devoted to it. It's just relevant here as showing you an example where these variations in coat color are not due to differences in DNA sequences, they're not due to mutations in genes, but they're due to differences in the chemical methylation patterns that are on the DNA. So we've known for a long time that DNA methylation is very important in controlling activity of DNA. Later on, when we talk about genes and their expression, the level of methylation associated with genes gives us some idea of whether they are active or inactive in terms of transcription. But what had been assumed for a very long time was whatever methylation, whatever chemical modifications were happening to the DNA, that would all be removed in the process of developing in really the maturation of the gametes, so that all of the gametes would carry the same DNA sequences. And they do. But what it looks like is some of these methylation patterns actually persist through the germline, through the gametes, and then get transmitted to the next generation. So these differences in the coat colors here are, again, not due to variations in the DNA sequences, but rather changes in the chemical modifications that have been done to the DNA. And those modifications have been transmitted to the next generation. How that happens, there's still a lot of speculation. It's a very, very active area of research and many, many unknown answers to the questions at this point. Okay? But we'll come back and we'll talk about epigenetics in a lot more detail later on. Okay. This is the part I know everybody came here for today, and that's to tell you how to build your own chromosome. So that you can go home and play junior scientist and build your own chromosome. And you get the Nobel Prize in the process. Okay. So the main, if you want to build a chromosome, you really need to have three main parts. You have to have an origin of replication, so you can replicate the DNA. You need the centromere, so you have this attachment point, so those microtubules can attach and form that kinetic core, so the chromosomes can segregate properly through meiosis and mitosis. And you need telomeres, because on eukaryotic chromosomes you have linear DNA, and these telomeres have special structures that stabilize them and keep them from being degraded by these nucleases in the cell. So we talked about centromeres before. Again, the centromere, I know it's a little confusing, the centromere is a DNA sequence. It's the point on the chromosome where ultimately the kinetic core will form. That's where the microtubules will attach. The first centromeres were cloned and studied from yeast. So for technical reasons, a lot of the early molecular studies of eukaryotic DNA came from studies with yeast. Now, yeast has unusually small chromosomes. It has about, I think, 16 pairs of chromosomes. Its genome is only a couple times larger than that of E. coli. One disadvantage of working with yeast is the chromosomes are actually too small to visualize them with the light microscope. You actually have to use an electron microscope to see them. In yeast, the centromere, because the chromosomes are very small, the centromere is actually exactly the right size for one microtubule to attach. But in most eukaryotic cells, the chromosomes are much bigger. One microtubule is not going to provide enough force to be able to move the chromosomes. So the centromeres in most other eukaryotic cells are much larger. But all they are is really a repeat of the same little small sequence similar to what you see in yeast. So these are short tandem repeats. So what do we mean by tandem repeats? They just mean one right after the other, sort of like cars on a railroad train. Okay? So, but we literally could have thousands of those because multiple microtubules have to attach to these chromosomes in these higher eukaryotic cells simply because they're larger. And you have to have the force necessary to be able to literally drag those chromosomes to the two ends of the cell. So the centromeres in yeast are maybe about 40 or 50 base pairs, whereas in, let's say, a mammalian cell, they're more on the order of about 100,000 base pairs. But again, it's just a tandem repeat of this short little sequence. The other thing is the telomeres. So there was a lot of suggestive evidence that there had to be something special about the structure of the telomeres. It was known from the chromosome mechanics who were making chromosome rearrangements in Drosophila. So they put the Drosophila in X-ray machines and bombard them with X-rays. And these X-rays do a really nice job of just slicing through and breaking the chromosomes. And then those broken ends would just knit together without any real rhyme or reason to it. So I said that these broken ends of DNA are very unstable. But if the DNA is linear in the eukaryotic chromosomes, there has to be something special about that end of the chromosome that's protecting it from these nucleases. Then there was another problem. We won't get into it yet. We'll get into it in chapter 12. But because of the anti-parallel orientation of the DNA and the fact that DNA polymerase only works in one direction, there's a problem with replicating the very end of a linear molecule. Right? If you know a little bit about that structure, you'd actually leave a single-stranded structure. That's not going to work in cells. Single-stranded DNA is very unstable, so you'd end up shortening the chromosomes at each cell generation. So there had to be some mechanism to be able to stabilize those ends of the chromosomes. Yes.
So if it's getting shorter with each replication, if that would cause aging.
Okay, jumping ahead. Yes. So this telomerase that we're going to talk about, it actually the chromosome. These telomeres are very dynamic. So in young, actively growing cells, tissues, and such, the telomerase is active and the telomeres are getting longer. Then as they go into quiescent homeostasis, the telomerase shuts down, then the telomeres start getting shorter. In many cell types, when the telomeres reach a certain critical length, it's a trigger for apoptosis. So you do see this in aging. In fact, they saw this with Dolly the sheep when they cloned her. She was actually born with old chromosomes and started developing some old age symptoms like arthritis and such while she was still fairly young. When they looked at the telomeres, she basically had the telomeres of an old sheep, even though she was fairly young. And as I sit here feeling my telomeres getting shorter by the minute. Okay. So there were a lot of ideas kicked around for a number of years about hairpin loops, similar to what we saw with RNA structure and such. It was really the discovery of this enzyme complex called the telomerase, and that this is actually an RNA protein complex. We'll talk about it in more detail later on. We talk about DNA replication. I think it's when we do that, but we'll talk a little bit now. And so it's an RNA protein complex, but its function is to basically keep extending out the ends of the chromosomes because of this replication problem. So we can extend the ends a little bit, trim them back a little bit, make them double-stranded, but it keeps the telomeres from getting shorter, it keeps the chromosomes from getting shorter. Like so many other things, the structure is the telomeres are a short repeating structure. So they're typically some number of N's or C's, more than 2, and then you have an A or a T. So it's sort of a CA, CT kind of repeat structure. It depends on the species and such. But you don't have to remember the exact sequences. In humans, it's CCC, TAA. And so here's what's happening at the ends of the chromosome. Here's the 3' end. And if you know a little bit about DNA replication and about primers, you realize there is no polymerase that's known that could just start at this point and fill in this gap. So what the telomerase is really doing is it extends out the ends of the telomeres, and it's a very dynamic structure. So it's literally going to add DNA sequences to the end. So as it shows in here, these sequences can vary anywhere from 2,500 to 1,500 times. And it's, again, it's a very dynamic structure that changes with age. It's species dependent, tissue dependent. But it's really this enzyme which keeps building out the chromosome and prevents it from getting shorter from one replication cycle to the next. So it was really from these studies that the 2009 Nobel Prize was awarded to Elizabeth Blackburn and Jack Szostak and Grider. Jack Szostak, I think, built the first artificial chromosome when he was just a graduate student. But it was really their work on this telomerase structure. The telomerase, as we talked about before, its potential role in cancer. So at a very fundamental level, cancer is basically uncontrolled cell proliferation. What you find in virtually every cancer cell type that's been looked at, the telomerase has been reactivated. So the telomerase has been a target for a lot of work in trying to find anti-cancer drugs. The idea being if we can inhibit that telomerase, then maybe we can get regression of the tumor cells. So the part that everybody's been waiting for is how you build a chromosome. So once you have all these little pieces, and this is what Jack Szostak showed, once he had an origin of replication, he did this with yeast chromosomes first. So he had the yeast centromere, because it was cloned there first. The yeast telomere was actually isolated from a protozoan first, but then shortly after that from yeast, and an origin of replication. Then basically you could just fill up the rest of the chromosome with whatever sequences you wanted. So initially he just used multiple copies of that bacteriophage lambda. So lambda we talked about as being that temperate bacteriophage that can infect E. coli. Well, its genome is a linear double-stranded DNA, about 50 kilobases. And he just showed that I can make the rest of the chromosome out of lambda DNA, put it into a yeast cell, and it functions as a normal chromosome. Actually, the larger the chromosome, the more stable it was. The other thing that came out of that was, oh, well, this will be very useful for molecular biology purposes, especially in things like the Human Genome Project. For technical reasons, it would be easier to work with much larger pieces of cloned DNA than you could do with plasmids and some of the other vector systems. And these artificial chromosomes would let you start working with DNA that was close to 1 million base pairs in length. So you're starting to get more than an order of magnitude larger. And just the larger the DNA you could clone, the fewer the clones you had to deal with. It's just a technical improvement that was really helpful in facilitating the Human Genome Project. Yeah, which is basically what's on here. Okay. They're really quiet today. Makes me suspicious. Like something's going to happen.
We're all thinking about the test.
Okay. Really? Thinking or just worrying?
Worrying.
It's easy. I got a 73 on it. So, well, that was with the curve. Okay. All right, here's where things get a little bit crazier. A lot of what I'm going to tell you about was first known way back in the 1960s, but it's only been about 3 years ago that some of the, even though we knew the structures were there and we knew the DNA sequences were there, nobody knew why they were there up until maybe about 3 years ago. And I don't think this is actually, I don't think that part, that EDGE project is actually in your textbook. All right. If you look at the genome of a prokaryote, you take E. coli, you got this circular genome. It's got about 6,000 genes on it. Genes in the conventional sense that they either code for an RNA like tRNA or ribosomal RNA, or they code for a protein. And there's very little other DNA that seems to be extraneous. So it's a very highly conserved genome, very efficient, no excess DNA. And because of the way bacterial cells grow, a little extra DNA baggage actually works against the organism. So these plasmids that these bacteria have, for instance, that carry an antibiotic resistance. If the organism is grown in the absence of the antibiotic, they very often lose that plasmid because there's no selective advantage to keeping it. And losing it just gives that cell just a slight advantage over the other cells, and they're growing under this very competitive environment. We don't really have that in eukaryotic organisms. But when they started looking at the structure of eukaryotic DNA, it was very different than what they saw in these bacterial sequences. So the first thing you need to think about is something called the C value. All the C value means is how much DNA is in a haploid cell from this particular species. We can measure it in base pairs, we can measure it in picograms of DNA, however we want to measure it. It's just the amount of DNA that's going to be in a haploid cell. So the C value for a human cell is about probably around 2 billion nucleotides, something like that. So a Drosophila cell has about 35 times more DNA than an E. coli cell. Okay. And human cells have about 10 times more DNA than a Drosophila cell. So we have 10 times as much DNA as a fruit fly, but we only have about twice as many genes. To a certain extent, that makes sense. The idea being, okay, as you kind of go up this evolutionary ladder, and you got more developmentally complex organisms, and we'll, to satisfy our vanity, we'll put humans at the top of that developmental ladder, it makes sense that the genomes get larger. However, there's some situations that don't seem to make sense. There's some salamanders whose genomes have 20 times the amount of DNA that a human cell has. There are some species of salamanders that, if you're not an expert in this area, they look like the same species, but they differ by at least tenfold in the amount of DNA that they have. A lot of plants have huge genomes. If I remember correctly, I think wheat has 10 times the amount of DNA of a human cell. So it gets us into something called the C-value paradox, which just simply means there are numerous examples of organisms that have a lot more DNA than we would expect them to have, and we don't know why. So it's just a paradox. That's all we mean by this C-value paradox. In general, as you have more environmentally, as you get more highly complex, higher eukaryotic organisms, the genomes get larger. But there are many anomalies to this, and we just don't understand why. But a big part of it is right here, that eukaryotic DNA has complexities to it. We'll call it complexities. I'll tell you what I mean by that in a moment, that you just simply don't see in bacterial genomes. Okay. Now, to get at this answer, we have to look at something, really take advantage of a biological property of the DNA. That is, we talked about how double-stranded DNA is more stable than single-stranded because those base pairs are hydrophobic. They don't want to be in the water. So forming that double-stranded structure where the base pairs are on the inside of the helix excludes water from the bases. So that's actually, thermodynamically, the single-stranded DNA will renature or find its complementary sequences spontaneously without any added input energy because it's more thermodynamically stable. So we can take advantage of this doing denaturation and renaturation experiments. For DNA, what do we mean when you denature proteins? Okay, you've probably all seen that the commercial, with the egg in the frying pan and your brain on drugs and that sort of thing. Is that a reversible process? Not usually. Okay, so most proteins, once you denature them with heat, chemical denaturants, or whatever, they do not reform into an active enzyme structure. What we mean by denaturing DNA is we're simply going to break the hydrogen bonds that hold the two strands together. in principle, the phosphodiester bonds, these covalent bonds, are not going to break. We're just going to break these hydrogen bonds. So we're going to denature the DNA, and then we can allow it to renature, which simply means we're going to take the single-stranded DNA, let it find its complementary sequences, and reform the double-stranded DNA. So it's a fully reversible process. There's several different ways to break those hydrogen bonds. The easiest way to do it is just with heat. So if you take a sample of double-stranded DNA, you isolate DNA, from human cells, put it, so you got it in solution. It's really it's rather soluble. Put it into a boiling water bath, heat it up to 100 degrees C, and you will denature the DNA. A very important idea is something called the thermal melting point, or the TM. This is the temperature at which this particular double-stranded DNA molecule is 50% single-stranded, 50% double-stranded. Because the DNA is just not going to fall apart, right? Which bases are probably going to separate first? The AT base pairs, because they have two hydrogen bonds. It's going to take more heat, more energy to break the GC bonds. Okay? So this thermal melting point depends on the GC content of the DNA. That's that G plus C that we talked about back with Chargaff's rules. So the more GC base pairs you have, the more stable and the higher the thermal melting point will be. All right, so just kind of in a cartoon form, you start out with your double-stranded DNA. As you heat it, it becomes single-stranded. And then under the right conditions, and you got to have the right salt conditions and such, you can slowly cool this back down, and it will renature and reform the double-stranded DNA. And this process is really at the heart of most of the molecular biology techniques, the cloning techniques, and such that have all arisen over the last 30 to 40 years. All the cloning sequences, making recombinant DNA and such, are all built on taking advantage of this natural property of double-stranded DNA. So we can do something called hybridization, which is exactly the same thing, but we're going to create hybrids. So we can take DNA from different sources. I can take human DNA and vampire bat DNA. They're going to share some sequence homology, and I can bring the single-stranded human sequences and the vampire bat sequences together and form double-stranded DNA, but I in fact have made recombinant DNA. So that's where the term recombinant DNA comes from, is that you're taking DNA from different sources and recombining them into a similar molecule. So most of the cloning work we'll do in the biotech lab and such, we make recombinant DNA because we're taking DNA sequences from Drosophila, vampire bats, whatever we want to use, and we're joining it to bacterial sequences. Okay. So this next part is on these different types of DNA sequences, and at the time, I think this will be a good stopping point. So everybody can wake everybody up, get them awake, okay? So we'll finish up chapter 11 next Monday. You'll probably be a little more focused because you'll be past the hurdle on Wednesday. So I'll use the remaining time to answer questions, and I'll go ahead and shut this all down. I'll put up